Good afternoon, everybody. I'm Christina Johnson from Door County Library, and I'm the branch manager at the Sister Bay Liberty Grove Library. I'm also a member of the Door County Reads Committee, and this year's book we have Braiding Sweetgrass for Young Adults, the adapted version of Robin Wall Kimmerer's book. It's been widely shared throughout the community here and our schools, and our series of related events that go with this book are focused on First Nation speakers, and it continues throughout all of February. So we have a lot going on. Right now, we just had our kickoff, a couple events this weekend as well, and a couple more things coming up. And then all throughout February, be on the lookout for publicity about that, including our author talk on February 1st. Today's event, though, we are here for the Finding Purpose in Research from an Indigenous Perspective presentation with a longtime friend of mine, Dr. Kat Milligan-McClellan. And thank you to everyone for sharing this afternoon with us. We have watch party locations here at the Sturgeon Bay Library and at the Bailey's Harbor Library, so thank you for anybody there in those rooms. And also, for those of you joining us online from home, we have quite a few of you. Just as a reminder, what we'll be doing is during the presentation, we'll have you remain muted unless prompted by Dr. Kat or myself. And you can use the chat box to share comments and questions. And while we can't promise that we'll get to all of them, we certainly will try to cover each question, and we thank you for your participation. A note also that this event is funded in part by a grant from the Wisconsin Humanities with funds from the National Endowment for the Humanities and the state of Wisconsin. Any views, findings, conclusions, or recommendations expressed in this project do not necessarily represent those of the National Endowment of the Humanities. We would like to provide our land acknowledgement from where we are broadcasting at our libraries here in Door County, Wisconsin. The Door County Reads Committee acknowledges the original peoples and primary caretakers of this peninsula from time immemorial: the Potawatomi, Menominee, Ho-Chunk, Odawa, Ojibwe, Petun, Huron, and Sauk peoples. The land we stand on today is part of the ancestral and historical homelands of each of these nations. We pay respect to these roots and connections and the traditional ecological knowledge that is present and vital today. May we appreciate those who came before us and commit to partnering with and learning from First Nations today and into the future. We are honored to share this time together. A little bit about today's speaker, Dr. Kat Milligan-McClellan. Dr. Kat is an Inupiaq and a runner, mother, and microbiologist, not necessarily in that order. She was raised in a remote community that was 80% Alaskan Native and above the Arctic Circle. She studied microbiology in the lower 48 for 20 years. That's when I learned her, that's when we first met. Dr. Kat earned her Bachelor of Science in Medical Microbiology and Immunology and her PhD in Microbiology, both at the University of Wisconsin-Madison. She uses stickleback and Alaskan fish to study host microbe interactions to determine the extent that the host environment contributes to host microbe interactions. As an assistant professor of molecular and cell biology microbiology at the University of Connecticut currently, she specifically examines how environmental toxicants that affect Indigenous communities affect the microbiota and development of the host, with a focus on host and microbe evolutionary traits and interactions. She also studies how to make STEM education more inclusive with a focus on improving the environment for Native students. Everybody welcome Dr. Kat.
Thank you for that. Welcome. I really appreciate that, Kuyana. Can you see my slides? Yeah. Okay, great. Perfect. Uvla lotak, uvana atagan napaktuk. I'm Dr. Kat Milligan-McClellan. Inupiaq miaruna, asin napaktuk miaruna, asin kikik tagrak miaruna, aglan inyunyaktuna, Connecticut me. Anaga Sally McClellan, Apaga James McClellan. I'm Dr. Kat Milligan-McClellan. I'm Inupiaq from the Napaktuk tribe, which is on the Nortek River just above the Arctic Circle. I grew up in Kotzebue, which I'll tell you about in just a little bit, and now I'm at the University of Connecticut. And I'm going to tell you today, my parents are Sally and James McClellan. We always talk about where we come from and who we come from, who our families are. So I'll be talking about my parents a little bit more in just a minute. I appreciate the invitation to speak here because this book is actually one of the books that really influenced me during my postdoc to think about how I could use my education to give back to my community. And that's been a driving force of the work that I've done ever since then. And reading this book really opened my eyes to the ways that um, Native Americans are trained in the educational system here in the United States, and what we can do to ensure that we're using this education for something that benefits our people. I always start with a land acknowledgement, but one of the things I've been doing lately is actually indicating to people that they should look at this landgrabct.org website. This is a website that our students put together, the Native students here at University of Connecticut. What I really like about it is that it shows not just whose land we're on. We're on the land of the Mohegan, Mashantucket, Pequot, Eastern Pequot, Shakatakoke, and others. But it also shows where the funding to start the University of Connecticut came from. And a lot of that funding came from the sale of lands in other states, land that was owned by Native Americans. The sale of those lands was used to fund the University of Connecticut and most land grant universities. So I'd like to send people to that website because there's so much information about not just whose land we're on, but also how land grant universities were funded. Today, what I'm going to be doing is I'm going to be telling you about where I came from and about the history of my family on my dad's side mainly. And then I'm going to be telling you about how I got to the place where I am now, the University of Connecticut, but also the way that I do research as an Indigenous person and how that was shaped by different events in my life. So let's start with where I came from. I'm from Kotzebue, Alaska, which is formerly known as Kikik-Tugrik, Alaska, or Kikik-Tugrik. It's just above the Arctic Circle. Our region is the size of the state of Indiana, and I like that I can tell that to this group and people will understand because not everybody understands how big Indiana is, because not most times when I give this talk, it's not to people from the Midwest. But our region is the size of the state of Indiana, but located above the Arctic Circle. The largest town in the region is Kotzebue, which is the picture that you see here. That's the biggest town. And you can see that it's relatively small. We have about 3,000 people now. It was 1,800 when I grew up, 80% Alaska Native, as Christina mentioned. And the people who live here, many of the people who live here still still practice subsistence hunting and fishing, which is basically using the land around us or working with the land around us to provide our food. So we fish in the water that you see here. There are many other animals that we use for food from that water. That land that you see around Kotzebue is filled with caribou and bear and other animals that we can trap for food. And then there's tundra all around this area that provides berries for us and medicines that we use. The other thing that I want to point out here is that our people have actually been in this area for 10,000 years. I like to stress that because we tend to think of the history of people in relatively short terms. The United States has been here for 250 years, roughly. It was colonized by people outside of this area some 400 years ago. And people tend to think that that's a really long time, but I'm talking about 10,000 years of history, 10,000 years of living with these waters, these lands, living with these animals, living with these plants, understanding how all of this works together and how things change from year to year and season to season. I think it's important to point that out right now because I think that gets overlooked when people think about how long Indigenous people have been on these lands. And specifically for my area, we have archaeological, linguistic, and oral history that shows that we have a really tight connection with this land. I grew up around this, knowing that we have this long connection, but not fully grasping it. The other thing I want to point out about growing up here is that while my dad was a subsistence hunter and fisherman and commercial fisherman, and I'll talk about that in a little bit, really my job was to focus on education. And so while I grew up helping my dad with fishing and hunting, a lot of that was actually done by my cousins, and then I was expected to go off to college, which I'll explain why in a minute. The photo on the right is a commercial fishing net and subsistence fishing net that my dad threw in one of the times that I was in Kotzebue. I love this photo because if you look really closely, about a third of the way up the net, there's actually a fish that's caught, and so you can see a little splashing of water. And it's one of my favorite things to do with my dad. Probably my favorite thing to do is to go up on the Noatak River, and I'll show you a picture of what that looks like in a second. As I said, we've been on this land for 10,000 years, but just in the last 2 or 3 generations, we've had monumental changes. So what I'm showing here is a photo of Mayadata, that's my dad's dad, in front of a house that he built with his own hands. He's got a dog sled team there. The dog sled, you can just make out, it's right next to the house, and that's a dog sled that he built. My dad also builds dog sleds, so this is something that's been passed down in generations. So you can see that he lived very closely with the land. He's got some fish drying on the roof of his house, and that dried fish would help feed on him and his family and his dogs year round. And then we also had caches of food that we built into the tundra. So we would dig large holes under the ground into the permafrost and store food there. And then we also had caches up above that were on stilts to keep the animals away, and we would store some food there. I like this photo of my dad, who is the very smallest baby in this photo, and his brothers and sisters and his parents, because it shows part of the shift to Western living. So my Anna, who's on the very far right there, my dad's mom, is wearing a parka, a traditional parka, but it's covered with cloth. And that's because by the time this picture was taken, we had a store in Kotzebue, and people were trading furs for cloth. And you also note that some of the people in this photo are wearing traditional skin boots, like my dad's dad, my Atata, on the far left there, and others are wearing boots that are more Western. So this is a photo that kind of indicates that even in this generation, we're seeing shifts towards a more lower 48 kind of living. I love this photo. This is not my dad, this is my uncle's, so it's my dad's generation. But I love this photo because my uncle is standing in a boat that my Atata built, made out of wood, covered in skins. And this is a boat that would be used for whaling. He'd be able to fit like 8 to 10 people in here. And he's holding a whaling harpoon, but then he's also wearing these absolutely stylish cloth coveralls, cotton coveralls, with his fabulous hat and his sunglasses, just looking fly. I love this photo. And then there's me, my generation. I am not driving a dog sled. I'm on a snow machine. I've got a traditional parka on with cloth mittens that are actually lined with fur. So it's a mix of lower 48 influences, but also Inupiaq influences. And we're on the Noatak River, which I can tell because there's no buildings and there's a lot of trees. So this is showing this shift in just 2 generations from my Atata to me. A lot of things are different that you can't see in these photos, like my Atata spoke fluent Inupiaq and I don't. I'm relearning some things, and some things I barely remember from when I was growing up. I don't live solely on berries and food from the waters and caribou and whatnot, although I do have my dad send those to me because he's awesome like that. So there's these big shifts that we see. And this is how I grew up. So this is Kotzebue, just taken a couple of years ago. You can see that there's houses all over, and you saw from that overview that there are schools and hospitals. What I love about this, though, is that if you go up on the Noatak River, it looks just like it did 10,000 years ago, with very few houses. This is a photo with my dad on the right and my kids on the left. One of the things I like about this picture is that my dad, I can imagine, is looking over this land and thinking about how it looks just like he did when he was growing up. He's climbed those mountains. He's lived off of just about every plant that you see in this image, from the fireweed to the berries that you can't see, to the Labrador tea, which we call tundra tea. All of these things are things that have been relatively unchanged from when he was growing up. And then the reason I include the picture on the left is because my kids are relearning some of this stuff. When we go back home, we have family and friends who teach them about the different plants and the medicinal uses for them. When do you pick the different types of berries? When are the fish in different parts of the river for harvesting? How do we know when we've collected too much or too little? So this just shows that we're trying to keep that generational or that knowledge from one generation to the next. Now, I do like to stop here and acknowledge my privilege. I've talked a lot about my dad and my dad's side of the family, but I also have a mom. My mom is Sally. She passed a couple of years ago. She's German-Irish, and she's a teacher. She was a teacher in Kotzebue for 30 years. And that's how my parents met. She was a teacher in Alaska. She moved up there right after the '64 earthquake to replace somebody who did not want to be someplace where the land shakes for minutes on end. And she moved up to Kotzebue when I was a year old with me. I love this photo because I'm in my traditional parka on the dog sled. My mom is in a Western parka. One of the things that this reminds me of is the fact that my mom adapted to living in Alaska the way that animals adapt to changing climates and changing areas. She learned how to skin a seal. She learned how to preserve meat. She learned how to set the net with my dad. In fact, she was a commercial fisherman with my dad for a few years. She learned about the caribou migration. And one of the reasons that I think this is so important is because some of my knowledge I got from my mom, just as I got a lot of my knowledge from my dad. But I like to acknowledge my privilege because my mom was a teacher and was German-Irish, I was expected to go to college. And that was not true for a lot of my classmates. I had a lot of really talented, really smart classmates who were not expected to go to college. And in part, it was because their mom was not the white teacher. I was expected to go to college because my mom was the white teacher. And that meant that instead of spending all of my time hunting and fishing and chopping wood and stuff like that, I was expected to also do really well in my classes. And that leads me to my parallel education. Some of my education is very Western, meaning I got it from Western education systems, what we call Western education systems. So going to college, going to high schools and elementary schools where they spoke only in English, except for an hour a couple of times a week when we had INUPAC classes. I also had this parallel education in being Indigenous. So relearning INUPAC, going upriver with my dad and learning about the different plants and when the berries are ripe and where different types of berries are, learning about the different types of fish that have come through Kotzebue and what it was like when there were abundant beluga versus when the beluga were no longer abundant in Kotzebue Sound. So my entire life, I've had this parallel education system between Western and Indigenous systems. And that's pretty much how I'm going to talk about the rest of that's pretty much what I'm going to focus on for the rest of this talk. I'm going to be focusing on some of the ways that I was educated in the Western systems and some of the ways that I was educated in Indigenous systems, not focusing just on INUPAC educational systems, but Native American education systems in general. I'm going to skip over a lot of things, like I'm not going to talk about going to Cornell for a year in hotel administration. I'm not going to talk about going to community college at the University of Wisconsin-Fond du Lac, although that was really important to me being able to continue in college and also shape the way that I think of educational systems, because going to community college was a really good idea for me. And I think it's a really good idea for a lot of people. And it's something that we don't give enough credit for when we think about higher education systems. But let me first talk about this Western education that I got when I went off to college. I was an undergrad at the University of Wisconsin-Madison. My degree was medical microbiology and immunology. I took all the classes that you would expect. So I took philosophy and African American studies and English classes. But the classes that I really loved were my science classes, where I learned about the TCA cycle, and I learned how to do these assays that would help us identify antibodies that were present in samples. That was amazing to me, that we could take a sample from a person and identify specific molecules that were in that person at one time or another. And we could use that to figure out which proteins were there, or we could use other assays to figure out what DNA was there, or what RNA is present, and what these molecules are doing in our body. And you don't have to know what any of these terms are to understand what I'm talking about. Just know that I was fascinated by this idea that we could learn about things that are so small that we can't see them with the human eye, and we could figure out what they're doing by doing these things in the laboratory that indirectly told us what was going on. The thing that I'm showing you on the far right is called a gel, and I have to show this to people because the scientists will know exactly what this is. It's a way of seeing DNA and figuring out how big that piece of DNA is. And it's the very first gel that I ever ran. This shows you how much I loved science, because this was something that I've saved since 1998. I still have it in a folder in my office. Now, when I was at the University of Wisconsin-Madison, I did a lot of research. I worked in 3 different labs. The last lab that I was in, I actually stayed as a lab technician for 3 years, and I studied the genes and the environments that were needed for this gram-positive bacteria to produce toxins. You've probably heard of Staphylococcus aureus. It's a microbe that can cause lesions. It can cause lots of diseases in humans. I was trying to figure out when it made toxins, and toxins are really small molecules that can cause a lot of damage in our body. I'm going to skip over a lot of the undergraduate stuff because most of my undergraduate education was spent trying to assimilate into non-Indigenous culture, because I thought that if I took all of these things that made me different, all of the things that identified me as being Inupiaq, but like wearing my earrings or my utaklooks or speaking like I lived and grew up in Kotzebue, if I assimilated, if I got rid of all these identifying things, that I would be more accepted by the people around me, because the University of Wisconsin-Madison was primarily white. Out of the 40,000 students that were there at the time that I was a student there, only 250 were Native. And out of those 250, there were 1 or 2 Inupiaq on campus at any time, myself and Julie Beans. That was incredibly isolating. And rather than embrace who I was, I tried to be like everybody else. When I was a graduate student, I decided to switch to studying parasites. So I studied this parasite called Toxoplasma gondii. I looked at it under microscopes. I looked at it in mice. I did a bunch of parasitology, which is basically just figuring out what this parasite was doing in the host. One of the things I want to note, though, is that neither one of these diseases has anything to do with being Indigenous. Neither one of these microbes is really important to people back home. And when I would describe the work that I was doing, the people back home were like, it makes no difference to me. Like, you could study Toxoplasma from here to the end of the world, and it would not impact my life whatsoever. What are you going to do with this education that's actually going to help people back home? And that was something that people would say to me when I went back home. The other thing that happened during graduate school is I started embracing more of my Indigenous self. This is part of that parallel education. I had this educational system in Western science learning about microbiology. How do you figure out which microbes are there? How do you figure out what they're doing in a human, in a mouse? How do you figure out why they're causing damage to the human or the mouse or some other organism? But none of that was education towards my Indigenous self. And so one of the things that I did during grad school is I reached out to Indigenous groups like Wunkshik and ACES, and Wunkshik is actually where I met Christina, so it has a big place in my heart. These were 2 Native student groups, and what that means is that Native American undergraduates and graduate students would come to these groups regularly. We would have meetings. We would do things like host powwows. So this is a powwow with Martine, and that's my first child looking up at Martine at a powwow where she's learning about the music. She's learning about the different ways that natives in Wisconsin dress, learning about their traditions. And I went to these groups not just to learn about other cultures, but also to find a sense of community and hear from people who looked like me. And we were able to talk about some of the issues that were important to us, not just maintaining our identities and holding on to traditions like at this powwow, but also fighting for things that we thought were important for Indigenous people. So on the upper left-hand corner here, I have myself. On the far left, that's Erin Birdbear, and then Ty and Dana. And we were fighting to preserve effigy mounds. And if you're in Madison, you can actually walk around the campus and see some of these effigy mounds to this day. And part of the reason is because Erin Birdbear led a bunch of different groups to preserve these effigy mounds. In this particular case, we drew the effigy mounds on us, and then we jumped into freezing water for the polar plunge. And it was our way of saying, hey, these are things that people need to pay attention to. On the bottom right, we protested to implement Indigenous Peoples Day. And in fact, Madison, Wisconsin was one of the first cities to implement Indigenous Peoples Day, in part because Wunk-Sheik and the students in Wunk-Sheik walked from the campus all the way up to the Capitol to tell members of Congress why we thought it was important that Indigenous people are recognized. And then I also held on to my identity by teaching Alaska Native games. During grad school, I realized that by embracing my Inupiaq identity, I was much happier. I felt more whole. I felt like the education that I was getting was going to have a purpose. It reminded me of all the things that brought me to that university. Teaching these games to other people reminded me and taught others about the importance of these games. And with that came the history of those games. And with that came the history of the relationship that we had with the areas that we lived in for 10,000 years, because every game is tied to living in that land. So by being involved with ACES and Wunk-Sheik, I was able to recognize that I didn't just have to get a Western education and assimilate, but it was also important that I be able to hold on to my Native identity and to be able to share that Native identity with others. Now, you can't stay in grad school forever, although I tried to. I was there for 7 years. I eventually earned my PhD. After you earn your PhD, you have to do something called a postdoc if you want to stay in academia. A postdoc is basically where you get more training. So for my postdoctoral fellowship, I had been studying microbes that cause disease. I shifted to host microbe interactions, basically studying microbes that are beneficial for people and other organisms. I also worked on actively figuring out how to make campuses welcoming for Native students. I had learned a little bit of that from Erin Birdbear and Ada Deer and other people, J.P. Leary, other people, Ned Blackhawk, other people at the University of Wisconsin-Madison who were making a difference on the UW-Madison campus. But I knew that there were other ways that we could support Native students on primarily white campuses, where most of the students were white and Indigenous students were a small minority of that. I also wanted to figure out how to study things that were going to be important for people back home. So I looked at different diseases that are caused by microbes and focused on ones that involve host microbe interactions. What I found in my research, just literature research, just reading papers, was that there are a lot of diseases that affect Alaskan Natives and Native Americans at a higher rate than people who are not Native. Obesity, heart disease, diabetes, liver and IBD cancer, all of these affect Alaskan Natives and Native Americans at higher rates than non-Natives. And they're all correlated with changes in the microbiome, meaning that if you look at the microbes that are in the gut of somebody who is Native and obese and Native and has heart disease or Native and has diabetes, their microbes look different from people who are white and obese and white and have heart disease, et cetera. There was also an indication at this point that the Indigenous people who don't have diseases have different microbial communities, that their microbes even when they're not sick, have different microbes than people who are not native. And this could be due to a lot of different reasons, but at the time that I was getting into this, we thought it was genetics. So I thought, okay, I'm going to get training in microbiomes and evolution or genetics. So I was trained in Karen Gilliman's lab on the left, and on the right, Bill Cresco's lab. And as you can see here, just by looking at it, I am the only Native person in two labs that had a lot of people in them. So again, I was at a primarily white institution, this time the University of Oregon, working in two labs that were led by people who are not Indigenous and who didn't necessarily know how to connect the work that I was doing with issues that were important for Indigenous people. So because I was trained in a microbiology lab and a genetics lab or an evolution lab, the way that I approached my question was to think about how the host genetic background contributes to host microbe interactions. Basically, if you're a geneticist, all diseases lead back to genes. It's like if you're if you are a person who works with tools, everything, if you're a hammer, everything looks like nails. Same thing. If you're a geneticist, everything comes back to genes. So when I started doing this work, I was looking for genes that changed the way that some populations of fish interact with microbes compared to other populations. And I was thinking of that as a correlatory, as being similar to humans. So humans, we have different populations and we have different microbes. I thought, okay, I don't want to work with humans because I don't know if I can work with humans ethically. And at the time that I was starting this work, we didn't have good ethical standards for working with Indigenous people. So instead, I figured I would work with this fish. And specifically, I was working with a fish that's found in Alaska. So here's a map of Alaska. I was working with fish that are around Anchorage. I'm from Kotzebue, and you can see that here on this map. The fish that I was working with were found all over this region of Alaska in this box. This is the South Central region around Anchorage. The yellow line there is the main road that leads out of Anchorage. It goes north and south. And if you look at this map that in which water is blue and land is brown, and the mountains are green, you can see a lot of blue spots. Almost every single one of those blue spots is a lake that has stickleback fish in it. And these are the fish I was working with, stickleback. Now, the cool thing is we can take these fish that are in different lakes and look at them, and each lake has a different population. Each one of those lakes has different microbes. It has different depths. It has different fish, different birds, different human activity. So each lake potentially looks different from a microbiology and a stickleback point of view. So by looking at different populations of stickleback around Alaska, I was able to look at many different populations that potentially had different relationships with their microbes. And I was really interested in how different populations respond to microbes and how that relationship changes. On the bottom right, I have a picture of freshwater and anadromous stickleback. Anadromous basically means that the stickleback live part of the time in salt water, like the ocean, and part of the time in freshwater, like a stream. The freshwater fish are in fresh bodies of water all the time. They just live there. And I was interested in whether the anadromous fish that live part of the time out in the ocean have different microbes than fish that come from freshwater populations where they always live in a lake. Now, the other reason I was interested in working with stickleback is because I knew that they have a lot of genetic variation. So if you look at fish from one lake and compare their genes to the fish in another lake, they're different. A lot of them are the same, but there are some that are different, which means that we could figure out which genes might be responsible for these relationships between the fish and their host. Their evolution and development is thoroughly described. That just means we know a lot about them. They've been studied for well over 100 years, including in the Midwest. Some of the earliest papers actually come from Wisconsin and Michigan. They share a lot of pathways involved with host microbe interactions and immune response with mammals, including humans. So if I could figure out some differences between populations and fish, they might tell us something about how different populations of humans have evolved relationships with their microbes. And then you can get large clutches from a single cross. What does that mean? That means that if you squeeze the eggs out of a single female fish, that you can get over 200, which means that we could do some really big experiments in the lab where we had a lot of fish from one female who looked really similar to each other because they were siblings. The other thing I liked about working with these fish is they're so cute. So these are fish developing inside of the eggs. The eggs are clear naturally, and I'm just going up and down in the microscope here. You can see the eyes, it's those big bulbous things. And now we're looking at the tail, and you can even make out different sections of the tail, and there's a little bit of pigment, that's the black stuff. And you see that thing in the middle of the egg that's just kind of pulsing, that's the heart. So we can watch things moving in real time. That's a 3-day post-fertilization fish. So after I've mixed an egg with sperm, on the right is a 6-day post-fertilization fish. And you can see the eyes are darker, so they have a lot more pigment. There's a lot more black splotches, so there's more pigment overall. And if we zoom in a little bit, we can see the tail looks different. There's this big bulb, yellow bulbous ball-looking thing that it's developing around. That's the yolk sac where it has a lot of nutrients. And there's these river looking things on the surface of that yellow ball. And I'm going to zoom in just a second. There we go. We're zooming in on the microscope, getting a little closer. And you can see things moving around in the river. Those are cells. So we can watch the development of these fish. We can watch how they're changing from no pigment to darker pigment, from the heart rate in early stages to later stages. And we can even watch individual cells as they're moving around the surface of these fish in real time. Like, we don't have to do anything to the fish. We just put them under a microscope and we can see all these changes. So when I was a postdoc, I figured out how to kill all the microbes that were associated with those fish. And that means I could raise them germ-free or in the absence of microbes. And I could also raise them colonized, conventional fish. I could raise them conventionally, which means that they have microbes in them. And this would give me an idea about how microbes are affecting these fish. I also did a bunch of things with diet and genetics, and I did some fun experiments with chemicals with my buddy Erica there. But all of this was to figure out how microbes are affecting the fish and their development. And it all led to this one big paper in which we figured out that different populations of fish have different immune responses to microbes. And the second important thing that happened when I was a postdoc is I had my second kid. And that was important because it changed the way that I was thinking about development in the host, and it changed the way that I was thinking about studying host microbe interactions. Now, I was getting this education in microbes and hosts, but I was also earning an Indigenous education at the University of Oregon. I was really lucky to be brought into this group called the Native American Strategies Group by Tom Ball when it was first forming. The thing that we were tasked with was figuring out how to make the University of Oregon more welcoming to Native students. Because again, I was on this campus with, at this point, I think it was 17,000 or 18,000 students, so about half the size of Madison, but there were very few Native students. And the Native students that were there were telling us about facing a lot of microaggressions in their classrooms and not being treated well, and they weren't sure that they were going to be able to use their education at the University of Oregon for anything important. So we worked to figure out how to make the University of Oregon a more welcoming place for Native students. And that's kind of shaped the way that I think of the higher education system in general. So we did things like we hired a tribal liaison. Jason Yonker is still with the University of Oregon, I believe, but he was hired in part through the Native American Strategies Group. He is from the Coquille Nation in Oregon, so he's a local tribal member, and his job really is to figure out what tribes need from the university and what the university can offer tribes, and how to build relationships between the tribes and the university. That's something that I have held on to, an important lesson that I've held on to from the University of Oregon through to the University of Connecticut. We also did things like hold the longhouse under Native American stewardship. So here I am with Gordon Bettles, who is the longhouse steward for the University of Oregon. He's since passed on. But he was the person who made sure that this building that was built with materials from the tribes and was in the shape of a longhouse, which is a traditional gathering space for Oregon tribes, that the things that we were doing in the longhouse were in line with what tribes found important, and that would be respectful for tribes. And so we made sure that one of the things that the Native American Strategies group did was to make sure that he was maintained stewardship of this building. And then I also learned how to teach Native students about microbiology. So this is Bogside. It was a summer camp for Native American high school students. We brought the students to the University of Oregon and Lane Community College and taught them how to be students on campuses and prepared them for college level courses. In my case, I was teaching them introduction to microbiology so they could get an idea about what microbes were and why it's important to study them. That outreach to Native students is something that I still hold dear to my heart. Eventually, I left my postdoctoral position to become an assistant professor at the University of Alaska Anchorage. This was really exciting for me because I was returning home. And I had all this education that I had learned at the University of Wisconsin-Madison and the University of Oregon, and I brought it with me to the University of Alaska Anchorage. Now, one of the things that I did is I switched the way that I thought about interactions between microbes and their hosts. So before, I had the same slide up, but it said, how does the host genetic background contribute to host microbe interactions? And that's because I was working with a geneticist. When I started working with these fish in Alaska, I could actually see where the fish were evolving. I could see the lakes, and I could see which environments had more human activity. And I could see that some were deeper and some were shallower, and that meant that the fish went to different places when the ice froze over. And I could actually see how thick that ice was, which told me about how dark it was in the lake. And I could see which animals were hanging out around those lakes, which gave me an idea of how microbes might be introduced into that lake and how it might be different from one lake to another. So instead of thinking of genetics, I really thought about the fish in their environments and the history of those fish. So where did those fish come from? Or how long had they been in that lake? Or how many generations ago were they interacting with a different kind of fish, but then now that other fish is gone and now its food sources changed or something like that. So I really started thinking about these different populations and the environment that they were in, which is something that we've been doing with humans as well. Instead of thinking about the different genetic background, different genetic differences between different populations, we're starting to look at Indigenous people and non-Indigenous people in this more holistic view, where we think about the socioeconomics, just like how much money do they have? How stressed are they? What access do they have to hospitals? Things like that that might be changing their relationships with microbes. So in Alaska, we did a lot of work in the field. So we go out and collect fish year round. So in the top picture, we're in the summer, and the bottom picture, we're in the winter. And then we would bring these fish or their guts, their tissues back to the lab, and we would figure out which microbes are there and what are they doing. I'm actually going to skip over a lot of the next couple of slides because I didn't realize how much I would talk about my background. So I'm going to skip over a lot of the work that we did in Alaska, but basically we figured out that fish from different populations have different microbes. That's all this graph is saying. And then we isolated a bunch of microbes, and then we figured out how to expose fish to microbes and figure out how individual microbes are affecting the overall development of the host, how they develop behavior and their immune system. Basically, I just figured out that different populations of fish have different relationships with their microbes. We also started focusing on fish development more seriously because I was a mom. I had 2 kids. My postdoc had 2 kids. And one of the things that we noticed is that sometimes the microbes are altered. Maybe your kid gets antibiotics. That change in the microbes that are there can affect the development of that person or fish. And so we really started thinking about how changing the microbes early in development affects later development. So I'm going to skip over that, but that's basically what we did here. And we tried to figure out how that was different between populations of fish. And overall, we figured out that, yeah, different populations of fish have different relationships with their microbes, and these microbes affect different pathways differently. So your eye might be different in one population versus another, but when you take away the microbes, it'll affect one population but not the other. Same thing for the swim bladder and overall growth. But the other thing that I did at the University of Alaska Anchorage that I brought with me to the University of Connecticut is thinking about how I can use this education to answer questions that are important for people back home. Development and microbes are really important for Indigenous people, but in addition to that, people back home, when I would ask them, what should I do with this education that I have? One of the things they said is, we want to know how different contaminants that are in the environment are affecting our fish and us. So we started focusing on how different ecotoxicants or contaminants affect the development of the host and how it does that through the microbes that are in the gut. We've looked at a lot of different contaminants. Perchlorate is one that's found in defunct military bases around Alaska. Fluoridone is an herbicide that's found in our waters. Lots of antibiotics are found in Alaska. Microplastics is a new thing that our lab is studying in Connecticut. Crude oil is something that we started studying when we were in Alaska because we had these big spills of crude oil in our waters that affected our fish and other species. And so we looked at how crude oil affected the development of our fish, and then what happened when we cleaned up that crude oil. If we cleaned up the crude oil, if we took that crude oil away from the fish, did the fish develop normally? And we did that in a bunch of different populations and figured out that some populations were not affected by crude oil and others were. And that's work that Kelly Ireland is doing at the University of Alaska Anchorage still. And then we also isolated microbes that are resistant to all of these. Here I'm showing you crude oil. And we can take these microbes that are able to grow in the presence of these contaminants, give them back to the fish, and figure out if they can reverse the effects of the crude oil or the other contaminants. And the last thing I wanted to talk about is how I'm approaching research in general. I mentioned that we're starting to think about things more in the ecosystem, like how do our fish fit into this ecosystem? How do our microbes fit into this ecosystem? And how is that different from one population to another? And a lot of that is based on the work of Rosie Allegado, who has been really inspirational. She's been doing phenomenal things in Hawaii, thinking about how microbes in disturbed ecosystems are affecting the ecosystem, but also how do you restore that? And she's focusing on lands that are important to Native Hawaiians. So she's thinking about how do you use this educational system to directly help people in your area, your people in that area. So thinking about ecosystem health, I've been working with a bunch of different people to look at how stickleback are evolving in new bodies of water. But the fun thing about this is we're not just looking at the microbes, which is my role. We're also looking at which genes are changing and whether the diet is changing and how the immune system is changing. And we're doing this in lakes in Alaska, so not in Kotzebue, but closer to my home, to determine how evolution is occurring in these big ecosystems. Now, I'm Alaska Native. I'm an assistant professor. We make up less than, I think we make up 0.4% of all professors in the United States, even though we're 2% of the population. So the other thing that I've been focusing on is mentoring and ensuring that the next generation of Indigenous scientists are able to figure out how to use their education and their tool sets to help Indigenous people at a much earlier stage. And I'm doing that with ACES and SACNAS, and I cannot recommend these enough for other Indigenous scientists or for people who are thinking about how to support Indigenous scientists. Both ACES and SACNAS take donations, so just throwing that out there. And I'm also working with Alaska Native students specifically through the Caleb Scholars Program, in which we mentor Alaska Native students who want to use their associate, bachelor's, master's, and PhDs to preserve traditional relationships in Alaska. So they figure out how to use their education to support subsistence hunting and fishing and saving languages and preserving our ways. And I'm doing all this while being a professor at the University of Connecticut. So if anybody is interested in learning more about this, I'm happy to talk to people. I'm Napoktuk on most socials. That's my Alaska Native name, which is down here on the bottom right. And I also have my email address up there. The other thing I wanted to point out is that I've been able to do all this work because I have students who are phenomenal. I've mentored 60 people in my lab in 8 years. And these people have just done amazing work. And I've also had a lot of mentors at every university that I've ever been at, and we've gotten funding from a lot of different groups. So I just want to say thank you for your attention. Thank you to Christina again for inviting me into the Door County Library. I'm a huge supporter of libraries. I think it's so important that we have libraries that are doing this type of important outreach and work. And I'm happy to take any questions. I am looking at the chat to see if there's any questions, but people can also, let's see, you can raise your hands if you have any questions, but I'll also read some of the questions that are in the quote or in the chat. So, okay. Okay, you were able to see this, great. Perhaps I missed something when I was having trouble, but why wouldn't her people think her microbiology work could help them back home? I was working on microbes that weren't important to people in Kotzebue. Toxoplasma gondii, for example, the parasite I was working with in grad school, is not found in my region at all. When I was at the end of grad school, so around 2008, 2009, they found Toxoplasma in sea mammals for the first time in southern Alaska, but they don't really affect, at the time, they didn't affect animals in northern Alaska, which meant that we weren't exposed to them, which meant that I knew nobody when I was growing up who had a Toxoplasma infection or was affected by it. Staphylococcus aureus was an issue with wrestlers and basketball players because you have a lot of skin-to-skin contact. But other than that, Staphylococcus aureus was not even that big of a concern for people in Kotzebue. Tuberculosis was a bigger deal. And in fact, when I was thinking about what to do for my postdoc, I thought about studying tuberculosis because Alaska Natives have a much higher rate of tuberculosis than other folks. But I decided to switch to beneficial microbes. That was a great question. Thank you. What was the result of killing microbes? That's a great question. So when I raised fish in the absence of microbes, I thought they would be smaller, but they weren't. And we've done a lot of microbes where we've taken fish and raised them with microbes, without microbes, with specific microbes. Overall, they tend to be the same length, but their internal organs tend to be different. Like their eye diameter might be smaller, or their swim bladder might be smaller, but it's not the same from one population to another. So you might have a germ-free fish from one population and their eye diameter gets smaller when there are no microbes around, but then you have another population and their eye diameter is exactly the same. So one of the things our lab wants to do in the future is figure out why, and that means figuring out which genes are changing, which proteins are different, things like that. Let's see. Okay, so I think those were all the questions that people had. Let me see if
Looks like
anybody's raising their
Poppy
hand.
has her hand raised, I think.
Poppy, I'd be happy to answer your question.
Hi, Kat. Thank you so much. This has been so interesting. I'm actually getting in touch from England. I found you on Blue Sky, so that's how I found out about this talk, which is fantastic. Right at the beginning, you talked about there being sort of an expectation on you to go to university, and I was wondering, was that within your family or was that within your broader community? And I'd just be interested to sort of touch on that a little bit more, and the fact that others weren't expected to go to university. But if you go to university and you're taken out of your community, does that potentially lead to a loss of skills? I mean, you were saying, you're having to relearn things.
Yeah.
So that there's downsides and upsides to that, isn't there?
Yeah. That's a great question. So yes, being taken out of Kotzebue did have a very big effect. Actually, let me answer the first part of that first. I was expected to go off to college because my mom was white. And that was an expectation from other people in my family. It was an expectation of teachers. It was an expectation of pretty much everyone that I came in contact with. They were like, your mom's white, she's a teacher. You're probably going to go off to college because we know that your mom went off to college. Leaving Kotzebue to go to college in the lower 48 was incredibly hard. So I, at one point, calculated how often I went home. Because it was so expensive to go home, I went home on average every 3 years for the 20 years that I lived in the lower 48. So I was in the lower 48 for undergrad, grad school, lab tech, postdoc. And all in all, it was 15 or 20 years. I can't remember now. No, 17. It was 17 years. It was a long time. And I only went home every 3 years. That means that I lost a lot of things. Like I knew how to gut a fish when I left. And when I came back, like my dad was like, oh, you gut fish for a living. Here, gut these. And I was like, I gut larval fish that are 5 millimeters. And you're asking me to gut 60 salmon. And I had to relearn how to do that. Skinning a seal. My mom would skin seals and walruses on the beach in front of our house. And I had a good idea about how to do that. I couldn't do it now. I would have a very hard time. I've watched videos. There are a lot of things that I've tried doing from the lower 48 to maintain all these, but it wasn't until I went back home that I relearned some of these. And when I was in Alaska, I also relearned how to do Inupiaq dancing. Some of my Inupiaq language is coming back to me after living in Alaska for a while and relearning it online, stuff like that. So I'm showing you the Caleb Scholars Program because one of the things that we're trying to do is ensure that the next generation has those ties. We give them funding to go home every year to maintain their Indigenous ways. So Caleb Scholars students, instead of going home once every 3 years, go home every summer. And we teach them things like, how do you gut a fish? We did that at the last Caleb Scholars gathering that I was at. We gutted fish together. We beaded earrings. We had somebody come in and teach us how to sharpen ulus. Like we are actively trying to ensure that this next generation goes off and gets their college education, but also maintains their Inupiaq traditions. That was a great question. Thank you.
Thank you.
Okay, Kevin says, hi, great talk. I hope this makes sense. I'm curious about your experiences and the support or not of your efforts to make education more accessible. Were institutions supportive or more naive to your efforts? For example, oftentimes universities will laud products of effort after the fact without supporting them. Are institutions becoming more purposefully supportive from the beginning? So one of the things that I think about all the time is how do we hire people and how do we keep them, especially faculty? We hire them based on how many papers they've written and how many grants they've gotten, and that's how we keep them too. When you're doing all the outreach that natives do to ensure that the next generation is able to attend colleges, we don't publish as many papers. Kevin's in my field. Kevin is actually a professor at another university, and he's in my area. He has published way more papers than I do. And part of it is because I'm part of the Caleb Scholars Program, and I've worked with ACES and SACNAS, and I've gone to those meetings and ABRCAM meetings, and I've mentored students through lots of programs. I've been part of SING and part of a bunch of other things. Each of those takes time away from my publications. So institutions, are they supportive? They really like the work that I'm doing. They would like me to publish more, and I get that comment all the time. Understandable, that's what scientists are supposed to do, but it makes it harder for me to justify giving time to natives when I know that the only way I can stay in academia to make this difference is to publish papers. Great question, Kevin. Okay, does anybody else have any questions? Okay, I am going to be hanging out here for a bit. So if anybody has any questions, I'm more than happy to answer them. I'm going to stop sharing my screen.
Okay, thank you, Kat. Just wanted to say, so moving, especially the photo of your dad and then your kids too. And oh my gosh, like I wanted to start crying when you first showed that. It's so sweet, and getting to see him. That's amazing photo of him, by the way, and just yeah,
Thank
reflective out there.
you.
It's so good. I'm glad that you captured that.
Yeah.
And also just so glad that you could join us in this series. When I was thinking of, who to invite, you instantly came to mind. Being somebody that reminded me a lot of Robin Wall Kimmerer, the author, being able to blend scientific and indigenous knowledge as something that you're like living and breathing and doing all the time in your profession. And just so glad you could kind of help be part of that niche and role in this series. So thank you for your
Yeah.
presentation and creating that, just a little glimpse of what you're doing and your background with all of us.
Yeah. I cannot explain enough how much her paper was inspirational, or I mean, how much her book was an inspiration to me, because at the University of Wisconsin-Madison, nobody asked me, like, how are you going to use your education to help people back home? It wasn't it wasn't stressed to me at all. And I didn't have any Indigenous professors that I could look up to in the sciences. People came like towards the end of my graduate career and stuff. But it wasn't something that a committee member would ask, for example. It wasn't something that any of the professors would be like, no, none of the professors said, how are you going to make sure that you're studying microbiology in a way that respects where you're coming from, but also is able to help people from where you're coming from. And Robin Kimmerer's book really opened my eyes to the fact that I could use this education to do that. It was a way for me to realize that I could use this education, but also that I could shape the educational system. I love the way that she teaches her students. She brings meaning into her research. She brings meaning into the way that she mentors students. She brings Indigenous ways into the way that she talks about her research at conferences and places like that. And that made me feel more open to doing that when I practice science. So it was incredibly meaningful to read this book and to think about how I could maintain my Inupiaq ways while being a professor, being a scientist, being a mentor at a university.
Her,
I see that Britt
oh, yeah.
raised their hand. Hi.
Hi. Yeah, I just wanted to say like really beautiful talk, and I just had a quick question. So I'm also a brand new PI. I just started as an assistant professor in 2022. So
Congratulations.
just from the perspective of another assistant professor, what are things that I can do to incorporate into my research environment or my lab or my teaching that can also help support initiatives like this?
That's a great question. So one of the things that struck me is that we, as professors, teach students who come from a lot of different backgrounds, and it's not always easy to figure out what's most important for those students. So for example, when I was in grad school, I had an aunt die, and aunts in my culture are basically like sisters. Like she was my aunt, but she was incredibly close to me. And it was hard for people at my university to understand why her passing was so impactful to me. And it was because she was teaching me Inupiaq words. And when I was pregnant with my daughter, I emailed her and asked her, what are the things that I need to do to ensure that I bring her in a way that's consistent with Inupiaq traditions, stuff like that. But people at my university didn't think of aunts that way. They were like, oh, your aunt is somebody that, you have Christmas dinner with or something like that, right? So that type of relationship wasn't as, they didn't understand that type of relationship. So I think one of the things that we can do as PIs is to think about where these students are coming from and do our own research into the types of into their backgrounds. So I don't think that we should be asking them, like, can you tell me all about your culture and what I need to know as your PI? I think it's important that we go out and do some of the research ourselves. I'm active on social media in part because it's a way to stay connected to INUPAC people from when I'm in other places of the world, but it's also because I follow people from a lot of different backgrounds who can tell me things like, this is what it was like growing up in as an upper class, highly or upper socioeconomic person in Wales, or this is what it was like to grow up in inner city Detroit, or this is what it was like to grow up in whatever area it is and whatever background it is. And by doing our own homework and figuring out where students are coming from, that gives us a better place of understanding what is important for them and how we should be interacting with them. I think the other thing that's really important is to acknowledge that not everybody who comes from a single background has the same experiences. So like I'm INUPAC, but if you met another INUPAC person, they would have a completely different experience. They would have different parents, they would have different upbringing, they would have different socioeconomic status, they would have lots of things that were different. So it's not like we can follow one person who is from India and know how all people from India grew up and how they interact or what's important for them, things like that, you know? So it's like, we have to do the homework, and then we also have to understand that and be open to communications from students. We have to be open to listening to them when they tell us what's important. There's so much more.
Yeah.
I think those are the important things, like being open and learning from other people and doing our own homework, things like that.
All right, thanks so much.
Yeah.
Kat, there's a couple comments that have come through in the chat.
Okay.
There's three
Kevin,
there.
hard to hear. Yeah, thank you, Kevin. How we generate impact. Yeah, that's an important question. How do we determine what is important to people when we're thinking about papers and grants and whatnot? How do we include outreach and mentoring students and things like that? It's something that we're thinking about at our university as we think about promotion and tenure and merit. And I think it's important that we have lots of people from lots of different backgrounds in the rooms when we have those conversations, because different people will give insight. Poppy, thank you. Yeah, I look forward to continuing to learn on Blue Sky. Yeah, I'm on Blue Sky, not Twitter right now. Can we access the recording? Tech is talking about that, and it'll be available on YouTube later. Yeah, I think those are all the comments.
Yep. Yeah. Anybody else want to sneak anything else in here real quick? I'm seeing another message pop in here. Yeah. From
Brian
Brian.
says, bonus of being a professor, my time is free whenever I want it to be.
Yes.
That's true. We
Yep.
have a lot more flexibility as a professor. It also means that sometimes you're working at midnight because you don't have a 9 to 5. But yeah, it does mean we have flexibility and free time, including attending things like this. So thank you, Brian.
Brian, we're excited. Yeah, please feel free to share with, you said you had several colleagues and your wife that would be interested in joining us, anybody else. We'll have these recordings up later in February and March sometime. If you follow our Door County Library Facebook page, we'll have posts there. We have a YouTube channel it'll eventually get processed onto. So yeah, that's great. And then, yeah, just really glad Poppy had mentioned both personally and professionally, this talk was very interesting for her in a university, you know, being able to support research and inclusivity. For anybody who maybe couldn't see the chats there.
Yep.
Yeah. So thank you everybody for joining us today, and thank you, Dr. Kat.
Yeah, I just dropped a link for I Was a Kid by Karen Romano Young. I noticed that she was on this. She draws cartoons. She interviews scientists from lots of different backgrounds, draws cartoons, and these are free to teachers. So if any teachers are interested in highlighting scientists who come from many different backgrounds, I highly recommend that. Thank you.
Thank you. Yeah, that's great. I think if one of our tech people want to take that link, we can even share it with some, like a recap of today's event. Be good.
Great, perfect.
Yeah, thank you. All right. Thank you, everybody. Have a good day.